Better believe 10 a.m. tomorrow at the front door. Let me tell you the story.
We're not running, are we?
Yeah.
Oh, sorry, I turned it off.
Going to see the brewing. The head brewer is mine's grad, right? Joe. Yeah. I will not be there today.
So you go Wednesday, Thursday, Friday, Saturday, Sunday.
So you're group 7? Who's group 8?
They're group 8? Are you we're 8, I think. Yeah.
You're next on the docket.
I have an exclusive test. He's a weirdo. Just talking.
Okay. Louis' Sweet Shop?
Lewis's Sweet Shop.
Lewis's Sweet Shop, okay. I'll keep it in mind.
If you want to turn off the
All right.
Turn off.
So what happens in my daughter's elementary school, they go like this. Somebody raises a hand up, everyone raises a hand, and everyone gets quiet. Alright, never mind. A little loopy with you days coming up here.
Are you going to participate in the festivities?
Are you doing the cake race?
Yeah, you have to.
No, I will not be doing the keg race. Introductions, please.
I'm Alexis.
I'm Sydney.
I'm Sammy.
Okay, so we did paragraph 7, and this is what it looked like. So our paragraph pretty much talked about the results of the GFP interference experiment, and they were looking at the effects of low doses and high doses of double-stranded RNA and how they affected the striated muscles and also the non-striated vulva muscles.
So they briefly mentioned a mosaic pattern, which we've encountered before in the previous section of the course, and we've seen it with the way that different cats express color on their fur. So some of the caliper cats, that's pretty cool. But the mosaic pattern is basically different versions of genes are expressed differently in different cells. And in this particular part of the GFP interference experiments, they had certain cells that didn't express the GFP, certain cells that did, and that was what they were evaluating when they went through and checked the dosage.
So as Sydney was saying, this paragraph kind of summed up the results of the GFP experiments. So they looked at a comparison of using high doses of the double-stranded RNA versus the low doses of the double-stranded RNA. So what they found was that the muscles that were derived in the embryonic, or in the embryo, so the GFP, it didn't change at all. And then the high dose, there was interference in all of the muscles. So it seemed to be that like the high dose of the double-stranded RNA was what worked best. But then in the non-striated muscles, no matter what the dosage was, whether it was high or low of the double-stranded RNA, there was no interference, so it didn't make a difference in those. So they pretty much found that the double-stranded RNA only affected the striated muscles. So, yeah. So when we were trying to figure out there's a sentence in there that says lone escaping cells, and if you Google that, the first thing that comes up is the cellular jail, which is actually a real jail in India.
So apparently people can get out of it. I don't know what that was. So, yeah. It's a character, but whatever. Any questions? It's pretty straightforward, yeah. What's up?
What's the differentiation between a low dose and a high dose? Like, how do they, in terms of concentration of doses?
I don't think they actually said.
They just said
it
low
just,
dose.
like, even in their tables it says
And
low
high
dose
dose.
and high dose. So I don't know that we actually know quantitatively what that was. Enough to kill.
Yeah, there's not a number. It's not quantified. It's qualitative. Also, if you're curious, this whole experiment is listed and cited in like 4 or 5 papers that I've been reading recently, so it's a big deal. And it was in the textbook.
Won the Nobel Prize?
I know. Other than the Nobel Prize thing. It's okay. No big deal.
Cool. Yeah, so thank you. So, why the difference between low dose and high dose is basically if you have an embryo and you put a small amount in here. So what we've identified, so this embryo, this fertilized egg is going to divide into 2 cells, and it divides and divides and divides. You wind up with a worm that's got 1,000 cells in it, just over 1,000 cells. So it's female, it's male, it's like 997. And that is, it may sound silly, but it's the same every time, okay? So if you put a low dose in here, when you get to 1,000 cells, there may have been a cell division where one of the cells didn't get a dose, didn't get its share of double-stranded RNA. So you'll express. So basically what they're saying is at the high dose, it knocked it out completely, and that's because there's enough double-stranded RNA to share, and every cell gets some, with the occasional lone cell somehow, you know, we talked about how every now and then a human cell might not get a mitochondria in it. So the cells divide. So that's the kind of stuff. Okay, that was it. Group 8. And I think group 9 is Katie and Orlando. And they have posted a video on YouTube, so I will be trying to find that. Did you see how good I am? They said go to YouTube and search Minds CSM Genetics, and it's one of those. Should be easy. Cheat sheet. What?
What? Oh. You want to see it?
No, thanks. Introductions?
I'm Karen. I'm Jana.
I'm Zainab.
So our paragraph was kind of more 3 mini paragraphs that were a couple sentences.
Sorry about that.
It's basically, they didn't know the mechanism of the RNA interference, but they do have 3 observations, so each of our mini paragraphs was one of the observations.
So the first observation was basically that if a dsRNA segment was made from an intron or promoter sequence, there was no interference detected after. So, yeah. And then, so basically, after transcription, there's no observable interference, but there's no conclusive evidence of what actually occurred in the mechanism before that. So
Basically, we can't tell what the actual effect on the actual gene is because after the transcription, which is the data they have, all that stuff is cut out, so anything could have happened with the stuff that got cut out.
And then table 1 just is telling us how the type of injected RNA affects the phenotype. And then the strong twitchers were trials where the effect was observed or interference did occur. Yeah, so interference only occurred when the sense and the antisense and the axon was injected versus the intron or promoter as mentioned previously.
Okay, the second observation was that the transcript that you use, or in the experiment they use MEX3, which is an abundant mRNA found in embryos and gonads of the C. elegans. So what they observed was that if you inject the dsRNA, it produces like a lot of elimination or decrease of the endogenous mRNA, which is like the mRNA found in the cells. It's kind of what we've been talking about in every group, that injecting the dsRNA causes a decrease or elimination in the actual transcript. So they found when you inject double-stranded, you get decrease, but when you inject just the antisense of the MEX2, and you get mostly retention of mRNA levels. And then figure 3 kind of shows the results of that experiment. So in A1 you have the negative control, what it would look like if there was no MEX3 staining. That's kind of what we're looking for in results to confirm our guesses. B is when MEX3 is just there. So it's all stained, it's all there. And in C, we see that the embryo from a parent injected with the antisense, so you're going to see a reduction, but not like you would see like in D, where you get the dsRNA injection.
Okay, so apologize, realized I didn't put a picture on here. Observation 3 was more to do with the offspring. So what they would do is they wanted to see how it could cross the cell boundaries, if it was just like, you put it in the gonads or whatever and that's where it stayed, or if it could go different places in the body. So they injected it in the parent head and tail to see if it would travel to the place, because it basically mostly affects the gonads and is where we see the interference show up. So they would inject it in the parent's head and then the interference would show up in the offspring, which was really cool because it showed that it could basically travel through the whole body without too much issue. And then the other experiment they did on this, testing the whole traveling cell boundaries thing, is they would inject it in the head of the adult and it would show up not only in the gonads but in all the other somatic tissue as well. And then the table 2, it was the third reference we had in our paragraph, and it basically just shows the results of this experiment, whether the twitching would show up in the offspring or not. That's our presentation. Any questions?
You hogged all the tables and pictures. I said you hogged all the tables and pictures. We got food.
Yeah.
You did great. Okay, thank you. Okay. So, let me see if I can find this on here. On this one, let me give a real short deal here. So we've got DNA, pre-mRNA, mature mRNA, right? Right? So here's the DNA. We've got promoter, and we've got +1, and we got all the sequences. Here, oops, sorry, we're going to get the pre-RNA, and we're going to have introns and exons in here, right? And here, we just have the mature RNA, messenger RNA, right? So basically what they were trying to do is they said, if I take sequences for an intron, so an intron is cut out at this stage, right, in the processor. If I have the sequence for an intron, do I shut down expression? So we know if I have double-stranded RNA for something that's in the messenger RNA, gene expression gets shut down. If I have double-stranded RNA for an intron, can I basically shut things down at this stage? That's the question they were asking in one of these experiments. And what they found is if you use double-stranded RNA for an intron, you express the gene. So they're trying to figure out where in the central dogma does the shutdown occur. And did they test promoter sequences too?
Yeah. Yeah.
And they said, yeah, so even the intron and exon stuff is here. What if we use a promoter? What if we get double-stranded RNA for a promoter? Will that shut down gene expression? The answer was no. So it looks like shutdown is not happening here. It's not happening here. It's happening to this thing somehow. Okay. So let me see if I can find YouTube. That's it? That's a channel. Yeah. All right. Hopefully we have sound.
Hi, I'm Alon Martinez. And please forgive us because this is really interesting video and research paper and everything. So bear with us. So we're here to talk about our section, and as many of you have probably said before, double-stranded RNA injections is a process that provides many tools and opportunities when it comes to defining specific functions of previously undefined genes. Genes that could not really be defined before by any other post-translational methods. And even though the effects of double-stranded RNA injections are potent and specific, there are also several observed limitations associated with it.
The first problem with this method is genes with similar or the same coding regions, they'll be affected. So it shows with the gene inhibition in multiple places, making it hard to identify the true function of the specific gene. For example, say you're looking at a specific gene with the coding region XYZ. Any gene that has that coding region XYZ will also be affected. So it'll show multiple effects throughout the organism. So it's hard to identify what that coding region XYZ truly does.
The second significant issue with this is that some genes have a very low expression in cells, and with low expression, it's going to be even harder to target these genes with double-stranded RNA injections. So because of that, these genes are going to have a much higher resistance to this, which also indicates that some or all genes and some or all cells will escape the effects, the desired effects of RNA injections.
So when this paper was written, and we're not sure any updated stuff, but when the paper was written, there were limited amount of organisms that they've done this on, including nematodes, other invertebrates, and potentially vertebrates. Some of the other studies have shown that this can also work in plants. Papers written by Harrison, Radcliffe, and Bolcombe, and another paper written by the Matsky brothers suggest that there are characteristics and structures of dsRNA viruses that are involved in the co-suppression, co-suppression genes in plants. So that is potentially promising.
All right, so hope this helped out and was as good as other groups. It was a little lot to make, but hope you guys enjoyed it. If you have any questions, feel free to ask Dr. Ogg right over there. Somewhere. Thanks.
Okay, cool. Unnecessary cuts. Oh man, that doesn't look good, does it? Okay.
Presentation, yay.
Any questions on that? There's one point in there. Yeah. And this actually kind of gets back to the group just before this. I'll do it at the end. Let's do the last group here. Last group on this paper, and then we have one final group coming up today. They were assigned and had the amazing opportunity to prepare an entire paper. You are not going to believe what they present. It's good stuff.
I lost it.
All right, group 10, introductions.
Hello. We are the final group. Pretty much what we're doing is our final
Introductions.
I'm Marcus.
And Allie.
Pretty much what we're doing is, this is the summary paragraph, so we're going to kind of go over what exactly has been happening this whole time. Very big. No. So I'm Marcus, again. What's that like? Okay, so we're going to start off. What is RNA interference? Pretty much RNAi is the knockdown of gene expression by small RNA fragments. And that essentially means like you put some RNA in there and it targets specific mRNA codes and destroys them, and it'll like inhibit gene expression.
So Fire and Mello proposed 3 different mechanisms for RNA interference. The first was a simple antisense model. The second was an RNA-based catalytic process. And the third was an RNA-directed chromosome remodeling. So in their first experiment, they proposed that inserting RNA into mRNA would cause twitching in the worms. However, when they performed the experiment, there was no effect on the worms. Their second mechanism included included the use of a catalytic component. So when they ran the experiment, they inserted 60,000 double-stranded RNA into the cell, and then the cell split into 1,000 progeny cells plus or minus. And this gave a ratio of like 60 double-stranded RNA to every 1,000 mRNA. Hence, a catalyst would be required to help the double-stranded RNA destroy all the mRNA. And when they ran the experiment, they saw that the mRNA was destroyed, which indicated that RNA interference was occurring and that the use of the catalyst was required.
And so in their third proposed mechanism, like Professor Ogg and Group 8 talked about up here, they were wondering if they can insert double-stranded RNA into the upstream of the gene, or like the promoter region, and see if that would affect initiation and elongation during transcription. However, when they did that, they found no effect, but later on, after this paper, or after they won the Nobel Prize, I think, it was found that you could go deep enough and actually affect, like, involve gene interference, and Professor Ogg will actually teach about that after E-Days. So pretty much what does this all mean? Essentially, RNA interference by double-stranded RNA is a potent and specific tool to regulate gene expression in eukaryotes. And here's a little picture if you guys wanted to look at it, but it just kind of shows different forms of RNA interference.
So then Byron Mello received the Nobel Prize based off of their discovery of RNA interference in 2006.
Any questions? This is a quick recap.
How come no memes?
I'm not good at making my own memes, so we thought we'd put a lot of pictures in there.
Cool.
All right, cool.
Before the final group comes up, thank you very much. I'm going to talk to you about, was it group 8? They injected it into the head of the worm. So if the worm is 1,000 cells in size, you got the head and the feeding structure, and the reproductive structures are down here. And basically, the worms are hermaphrodites. In general, most of the worms are hermaphrodites, meaning they have male reproductive parts and female reproductive parts. So the vulva and the testes are all in this area here. And so the eggs are manufactured in this area down here. So they injected the double-stranded RNAs into the head of mature adults. And they saw that cells that control the vulva were inhibited. More than that, and this is the really cool thing, is when this egg was dumped out and developed into an adult, that gene was knocked out in this progeny from this guy that was injected. That would be like me injecting you, and then you have a kid that is affected by what I injected into you. It actually turns out the way the system now works with worms, if you want to knock out expression of a gene in worms, you take a plate with media on it, and you grow bacteria on it, and these guys actually eat E. coli. And what you do is you make a plasmid, you put that plasmid into the E. coli, and the E. coli will produce a double-stranded RNA that is specific for the gene you want to knock out. The organisms eat the bacteria that have this double-stranded RNA in them. The double-stranded RNA gets transferred from the bacteria into the organism and knocks out the gene. Not just in the organism, but all the progeny from the organism, whether these guys eat the double-stranded RNA or not. Wow. So, if the elegans genome is 20 to 25,000 genes, I could knock out any gene from the elegans genome by creating a plasmid that makes double-stranded RNA. You guys are doing that. You guys are making plasmids for your genes. The technology you're using could be used to knock out any gene in the elegans genome. And if it works in elegans, what they're proposing is maybe it works in other organisms. And we have the same mechanism to shut this down. And I'll talk about that mechanism later. So in theory, we could make food. You could eat double-stranded RNA that has a specific sequence, and you could target some gene and knock that gene out of your genome. You could stop expressing that gene from your genome.
It would even survive with stomach.
Yeah. And we're going to see, that's a great question and a great segue. Why don't you guys come on up? This is great. You're going to love this. Can you introduce yourselves?
Hold on.
So my name is Ryan Learning, and this is Kate Sweeney, and we are presenting on the targeting of a specific protein based on an RNA sequence that was injected from the ingestion of food.
What year was your paper published?
It was 2011, 2012.
So, 1990. When was this paper? 98 to 06, Nobel Prize. 6 years after that, you're going to see this incredible discovery.
Okay, so we were already introduced. So then we were going to introduce some vocabulary. This paper is about cross-kingdom RNA or microRNA regulation, and before we only knew there was cross-species RNA regulation between human to humans or between all mammals, but now this paper discovered that intaking rice can also manipulate our gene regulation. So now we can have a discovered cross-kingdom gene regulation. It's about 30% of post-transcriptional gene regulation, and this is mediated through microvesicles. So you'll eat, for example, rice is what this paper is about, and all of your cells will intake it and then excrete some microRNAs in these microvesicles, and let me go to the next slide. And then what is supposed to happen is this LDL receptor, or any receptor, will uptake the gene regulator. Okay, so I want to get to a specific example. Okay, I'm using non-expert. So eating rice, there's high levels of this microRNA in Chinese people, and this is also abundant in rice. So they kind of wanted to figure out that relationship, and so Ryan's going to talk about the experiments they did, but I'm just going to give the conclusion pretty much. So this microRNA right here, MIR168, binds to our LDL, which is bad cholesterol receptor. So what the receptor does is it uptakes our bad LDL, and so that lowers our cholesterol. But what this microRNA does is it inhibits the production of this receptor gene. So it is not allowed to, or it can't further uptake our cholesterol, or our bad cholesterol LDL. So when we're eating rice, it inhibits the gene expression of the receptor, and therefore increases our LDL levels. Does that make sense?
Yes.
Okay, so going into the cross-kingdom that Kate briefly talked about, as she said, they first observed this in Chinese people, and obviously it wasn't a human DNA sequence, so they were looking for a way to find this RNA sequence in other groups. So the first thing that they did was they found these two sequences, and they decided that they were going to observe it in mice. So what they did is they found the microvacuoles and actually saw it, and were able to prove that it actually survives through the gastrointestinal tract and can go throughout the whole body and affect what is produced. So some of the testing that they did was a stem loop testing where they were able to take that sequence and prove explicitly that that was the one based on the other mRNA sequence that they've also put in. And then they also did an oxidation test where they tested the 2' and the 3' end, which also confirmed that this was not coming from humans and was being cross-linked. So the second type of testing that they did, obviously they proved that it was there. They needed to figure out what it actually did and if it actually affected humans in a certain way. So they found this gene, or the protein here, LDL wrap, and it is what wraps around your LDL proteins and actually allows your blood to be free of it and escape your body. So it's really good for you. And they did a northern and western blot on those to test the RNA sequence and the protein to see whether they were there. And then what they did is they took mice and fed them rice. And they put the antisense in and they did the sense to prove that it is actually happening and is what is causing the cholesterol to appear within these mice.
So, to wrap it up again. Okay, so eating rice will pretty much raise your bad cholesterol, the LDL, because it inhibits the production of your receptor gene. This is on a larger scale, though. This is much more important regarding the cross-kingdom gene regulation, and a lot more further research can be involved in that, or regarding that. Also, we're maybe realizing that we don't only need vitamins and minerals and stuff like that, but more novel nutrients coming from the food that we intake regarding microRNA. And this obviously is not just for rice, it could be for all of our corn, and maybe some epidemics or something could be happening, influencing these things. And yeah, that is it.
Do you guys have any questions?
Can I ask Dr. a question?
Sure.
Do you have any proposed mechanisms on how it survives the digestive tract?
Oh, yeah, we talked about that a little bit, but it's the protective layer that it actually puts around the mRNA as it goes through. It's just not able to break it down from that part of the body.
So with like the chains and stuff?
I don't know the details on it, but it seems like the digestive system has a way of recognizing nucleotides and taking them in intact before they get destroyed.
All of the other DNA and everything regarding that also wasn't able to get through the digestional tract, so it's definitely something to do with the
Oh, the rice packages it in a vesicle. That's what the MD was. The rice packages this microRNA in a vesicle, and that vesicle gets taken in. It's protected in the stomach, and then it gets taken in by the small intestine.
So what exactly did they observe, like in the Chinese people, that they had like high cholesterol levels, or what was the observation?
Yeah, so what they did was test how much of the LDL was present.
Okay.
And that's why they did the rice testing.
Okay.
So they did normal control, just feeding them with rice to test their cholesterol levels, and then the antisense to actually wrap the proteins.
Okay.
So the Chinese have a lot higher cholesterol than everybody else.
That's what happened when they removed rice from their diet?
Yeah, there wasn't,
Like they had it in their diet and then they just took it out?
Yeah, so gradually it decreased over time, but it's just like a natural rotting process.
yeah, the control was like a chow diet, so it was something that,
That was in mice.
yeah.
Not in
Not
Chinese.
Yeah.
Chinese.
I think that generation after generation, that it's pretty genetically printed in that, and so I don't think you could even run a test on the Chinese people, because at the end of the paper it said that it was pretty set in stone that the levels, so even, because, okay, so right after the mice were eating it for several hours, they could see a difference in their cholesterol levels, but I don't think that it would work on people.
So don't eat rice.
Well, it'll just raise your bad cholesterol. So.
Which we
So
don't really
other
know is
things.
that bad.
LDL
Yeah.
is for sure bad.
Well, yeah. There is a good cholesterol
It's just
and
the
a
ratio.
bad cholesterol. But when they're actually looking at that, it's just they're looking at the ratio
Yeah, and they're
and
just-
comparing fat people versus skinny people and saying that, oh, skinny people have this ratio, let's try to make the fat people have this ratio, maybe that'll make them better.
And this
But
was
we don't-
strictly testing LDL, so we don't even know.
Yeah, how it affects it.
Others.
Yeah. But it's not just, LDL does not simply equal fat. There's a lot more to that that we don't know. All right, applause, that was awesome. I want you to think about this.
Yeah, I also.
Rice, in its DNA, encodes for something that gets transcribed, and it might get processed. And that thing does not code for a protein sequence. The purpose of this RNA that gets produced, it appears the only purpose of this is to go into some animal that's going to eat the rice and modify its cholesterol levels.
That tricky rice.
Why? So you and me sitting here like, oh, I don't want my bad cholesterol, I'm not going to eat rice. That's not where we came from if you believe in evolution. You think 50,000 years ago, if somebody is consuming this, they may have a higher concentration of LDL in their body. And it may have been advantageous at that time because cholesterol was at such low levels. And those people at that time may have been having a hard time getting the nutrients they needed. That consuming the rice, which modified their physiology this way, may have given these people a selective advantage. Such that, or the people who were growing rice, the rice evolved to support that physiology. Does this make sense? So we're talking about this huge obesity problem here in the United States. And we're attributing it to corn sugar. And sugar and all kinds of diet things. What if there is a microRNA in corn that gets through the processing, and that as people are eating more and more corn, because that's what we grow, it's so easy to grow, that microRNA is being delivered to us and it's affecting our physiology. Maybe it's not the sugar that's doing it. Maybe there's this other new type of thing. Or both. Or both. I think it's probably the sugar. So it seems like probably long ago there was a mutually symbiotic relationship that slowly established between the things that ate rice. And rice is like, okay, go ahead and I'll provide you with this nutrient. You just need to make sure you plant me and propagate my genetics. So it became self-supporting. So again, like they were saying, you need to eat carotene, you need to eat niacin, you need all these organic molecules to help your enzymes work. You need to eat ions, because you need salt concentration to certain amounts in your body. Maybe these microRNAs are a necessary new type of vitamin.
Old type, been around long.
Well, yeah, newly discovered that we were unaware of until 2 years ago. Mind blown, right? Maybe not. With that, let's go eat.
Did you bring us rice?
So, the mechanism of this. So this final group, group 10, summarized, it looks like it happens at the RNA level. I want you to remember splicing. We had those small nuclear RNAs, what were they, about 20 nucleotides long? I called them guide RNAs. And you remember at the splice site there's that sequence, CAG-GTRAGT, right? Trag, CAG trag, right? And that defines one splice site, and then there was the CAGG was the other splice site. So proteins, you grab those 20 nucleotide RNAs, and they use them to find the splice sites, right? So these microRNAs, the reason that they're called microRNAs is they're really small, 22, 25 nucleotides long. there is a protein that binds that. And the protein then uses that. It separates the strands, and it takes one protein will get this strand, one protein will get this strand. And they go around looking for something that hybridizes to it. So, one machine will get this guy, and one machine will get this guy. This guy isn't going to find a target because it matches this sequence here. But this guy will find that target. This machine comes in and cuts this and destroys it. So that every time transcription occurs and you make a new messenger RNA, this guy's going to be there to get it destroyed. That's the mechanism that was revealed in this paper. There appears to be a different machine. I don't have a different color, so I'll do a machine. There appears to be a different machine that will bind the guide RNA, the microRNA now being the guide RNA. And what this thing does is it goes into the nucleus, and it looks for a messenger RNA in the nucleus that it can bind to. And if it binds to that, it follows that RNA, basically it goes to the genome where that sequence appears, and it methylates the DNA. What does methylated DNA do? It what?
Starts folding.
Starts folding up, and it recruits histone deacetylases. So you take the acetyl groups off, and now the histones compact the DNA. And then you get chromatin spreading, and you package all of this stuff in heterochromatin. MicroRNA not just destroys the messenger RNA, it goes to the source and shuts down transcription. And as we talked about, your DNA methylation pattern, you can pass that on to your kids.
Wouldn't that knock out other genes though?
Yeah, any genes nearby will get knocked out too. Everything on that radial loop on that rosette.
This doesn't help figure out how anything's alive.
Yeah, it only complicates.
It only makes
How can I be alive? How can I have eaten all this food and not had every one of my genes shut off every time?
Or at least one important one.
Yeah.
Like once in the millions of years that life has been going on. I don't know, it's weird.
Natural selection. It's evolved to be ideal, right?
It is pretty cool.
And selected to be that way.
It just makes no sense.
Okay. Thank you very much. Go drink beer, but not too much. Reasonable amounts.
No.